Dear students, welcome to EPG Pathshala. I am Dr. Meena Yadav from Maitri College, University of Delhi. I am going to discuss about the neuroendocrine feedback and response to varied stimuli. After studying this module, you should be able to discuss the hypothalamus hypophyseal portal system, the somatotropic axis and its regulation, the brain-pituitary-gonadal axis and its regulation, the brain-pituitary-thyrotropic axis and its regulation, the brain-pituitary-adrenal axis and its regulation, the lactotrophic axis and its regulation, the regulation of oxytocin secretion, the various factors influencing the brain-pituitary target organ axes. Hypothalamus secretes several hormones and all of them are regulated by some mechanisms. The feedback regulation of neurohormones can occur at various levels like at the gene transcription level, translation level, post-translational modifications or even at the release of the hormones. The feedback regulation can be broadly of two types, the positive feedback and the negative feedback. Positive feedback regulation. Sometimes the action of the hormone on a target tissue causes surplus secretion of hormone. For example, the secretion of luteinizing hormone. The action of estrogen on the anterior pituitary causes the release of more luteinizing hormone before ovulation. The secreted LH further acts on the ovaries to secrete more estrogen, which in turn stimulates the secretion of further LH from anterior pituitary. This action causes the heightened release of LH, which is known as LH surge, and it is an example of the positive feedback regulation. Negative feedback regulation. The majority of the hormones which are secreted by brain are regulated by the negative feedback mechanism. When the levels of a hormone increase in the blood, the hormones inhibit the release of themselves by inhibiting the secretion of releasing and inhibiting hormones from hypothalamus, and which subsequently inhibits the release of hormones from the pituitary gland. For example, the brain-pituitary-thyroid axis, the brain-pituitary growth hormone axis are examples of the negative feedback regulation. The secretion of hormones from the pituitary gland is under control of signals from hypothalamus. There can be two types of these signals, either hormonal signals or neural signals. The posterior pituitary secretions are under control of nerve signals from hypothalamus. These neurons originate from various nuclei in hypothalamus and extend their axons such that they terminate in the posterior pituitary. However, in case of anterior pituitary, the signals come in the form of hormones from the hypothalamus. These signals are called as releasing hormones or the factors, or inhibiting hormones or inhibiting factors, and they are secreted within the hypothalamus. They are carried to the anterior pituitary via a rich capillary network which is called as hypothalamic hypophyseal portal system, which is primarily located in the median eminence. These hormones from the hypothalamus then act on the specific target secretory cells in the anterior pituitary, and they cause the release of respective hormones in the general blood circulation. Hypothalamus is a center which collects the signals from all over the body and regulates the secretion of various hormones from pituitary to maintain the homeostasis inside the body. The control of secretion of hormones from pituitary is regulated by various pathways or axes which include the flow of signals in a loop including brain, pituitary and the target organs. So, there are 5 such neuroendocrine axes which regulate the growth, reproduction, metabolism, lactation and stress. Let us discuss all these axes one by one. So, the first axis is the neuroendocrine axis for growth, that is the somatotropic axis. The growth hormone promotes the growth of the body by influencing the synthesis of proteins, cell proliferation and differentiation. This axis is involved in regulating the growth of the organisms and works through the following hormones: hypothalamus, which secretes the growth hormone releasing hormone and growth hormone inhibiting hormone or somatostatin; anterior pituitary, which secretes growth hormone, which is also called as somatotropin; and the primary target organs, that is the liver, which secretes the insulin like growth factor 1 or IGF-1. There is a unique feature of this axis as growth hormone and IGF-1 both can produce the growth promoting effects either alone or together. However, both the hormones are required for the complete growth promoting function, as absence of any one of these due to mutations may result in dwarfism. The hormones secreted by somatotrophic axis are different in nature than the hormones in the other axes because the hypothalamus secretes GHRH, which stimulates the secretion of growth hormone from specialized cells in anterior pituitary, which are called as somatotrophs, and promotes the growth, and it also secretes GHIH, which inhibits the secretion of growth hormone and thus inhibits the growth. GHRH is secreted by neurons in the arcuate nucleus in the hypothalamus. This nucleus also responds to changes in the blood glucose levels and causes satiety if blood glucose levels are high, and hunger if the blood glucose levels are low. Further, growth hormone interacts with the GH receptors in liver and stimulates the secretion of IGF-1. The GH and IGF-1 have common target organs like bone and muscles and promotes their growth, regeneration and metabolic homeostasis. Growth hormone and IGF-1 both are a part of feedback regulation of somatotrophic axis. Growth hormone is released more during night as compared to day and is under hypothalamic control. In addition to GHRH and GHIH, ghrelin also controls the secretion of growth hormone. It is produced in the hypothalamus and stimulates the secretion of growth hormone. Although there are several factors which influence the secretion of growth hormone, but the most important factor is GHRH itself and not the somatostatin. GHRH binds to its receptors present on somatotrophs in the anterior pituitary and stimulates the secretion of growth hormone. The binding of GHRH to receptors causes the activation of adenylyl cyclase, which causes an increase in the level of cyclic AMP. Cyclic AMP causes an increase in the levels of intracellular calcium, which further causes the secretory vesicles to fuse with the cell membrane and release growth hormone into the blood. Cyclic AMP also induces the transcription of gene for the synthesis of more growth hormone. So, growth hormone regulates the GHRH secretion from hypothalamus by negative feedback mechanism, but it induces the expression of somatostatin gene also. Thus, if the GH is lacking or its levels are low, the somatostatin gene will not be expressed to produce somatostatin. IGF-1 also acts in the similar way as growth hormone and is a negative feedback molecule for GHRH and stimulates the expression of somatostatin gene. The GHRH neurons are located in the arcuate nucleus in hypothalamus and release GHRH in the portal capillaries in median eminence and other hypothalamic regions too, where it may control the feeding behavior. Thus, GHRH neurons may get stimuli from other neurons like in the central nervous system like alpha adrenergic, dopaminergic, serotonergic and encephalinergic neurons. The cell bodies of GHRH neurons are densely located in the arcuate nucleus, whereas cell bodies of somatostatin neurons are dispersed throughout the central nervous system. However, the cell bodies of somatostatin neurons which are involved in growth are concentrated in periventricular nucleus. Most of the neurons from hypothalamus have their axonic projections into the median eminence, but somatostatin neurons also interact with GHRH neurons and thus inhibit them. The next axis is the neuroendocrine axis for reproduction, that is the brain-pituitary-gonadal axis. The brain-pituitary-gonadal axis plays a very important role in the propagation and survival of the species. The gonadotropins LH and FSH control the growth of testes and ovaries and their further development and hormonal activities. The hormones which are secreted and play a role in this axis include the hormones secreted by hypothalamus and the preoptic area, which secrete gonadotropin releasing hormone GnRH, also called as luteinizing hormone releasing hormone or LHRH. Anterior pituitary secretes gonadotropins, that is luteinizing hormone and follicular stimulating hormone. Gonads, that is testes in males and ovary in females, secrete sex steroid hormones like estrogens, progestins and androgens. The GnRH neurons in hypothalamus extend their terminals in the median aminus and release GnRH in a pulsatile manner. The GnRH acts on its target cells in anterior pituitary, that is gonadotropes, and stimulate them to secrete gonadotropins, that is LH and FSH. These gonadotropins are secreted into the general circulation from where they reach their target cells in the testis and ovary, which further stimulate steroidogenesis and spermatogenesis in males and oogenesis in females. The binding of these gonadotropins on the receptors of target cells in gonads leads to the synthesis and secretion of sex steroids from the gonads, that is estrogens and progestins from ovary and androgens from the testes. However, the ovaries also produce certain amounts of androgens and testes produce estrogens and progestins. The sex steroid hormones, once they are released from the gonads, enter the general circulation. They bind to the receptors on the target cells scattered throughout the body. The target tissues, they respond by producing secondary sex characteristics and other adult reproductive functions. Certain regions in the brain have receptors for sex steroid hormones, including hypothalamus, and the interaction shows several effects on the central nervous system. The most important effect of sex steroids is in the feedback regulation of GnRH secretion from the hypothalamus. The regulation of reproductive axis changes as the function of the reproductive system changes over a period of time during development. For example, during the late embryonic period in the development or just after birth, the activity of brain-pituitary-testicular axis increases, while there is a less activity of brain-pituitary-ovarian axis. However, shortly after birth, the reproductive system goes dormant, maybe due to inhibition of GnRH or due to lack of stimulation of this axis. This stage is called prepubertal hiatus, where there are low levels of GnRH, gonadotropins and sex steroid hormones. In males, the sex steroid secretion and inhibition is controlled by negative feedback mechanism. The release of GnRH stimulates secretion of gonadotropins. LH further stimulates the release of testosterone, which in turn inhibits the release of LH by negative feedback loop. The main process by which testosterone shows the negative feedback on hypothalamus is by inhibiting the release of GnRH, and thus stops the further release of LH and FSH by anterior pituitary. This results in the inhibition of secretion of testosterone by the testis and thus completes the negative feedback chain. Whenever there is excess secretion of testosterone, this negative feedback ensures the levels of testosterone are reduced to normal levels by inhibiting the secretion of hormones at two levels. One is the hypothalamic hormones and second is regulation at the anterior pituitary hormone. Conversely, when the testosterone levels are too low, the hypothalamus secretes GnRH, which in turn stimulates secretion of LH and FSH and further secretion of more testosterone from testes. In testes, when there is low production of sperms within seminiferous tubules, there is increase in the production of FSH by anterior pituitary. Also, if there is too high production of sperms, the FSH secretion slows down. This phenomenon of regulation of FSH secretion is controlled by a hormone which is secreted by the Sertoli cells within the testes, which is called as inhibin. Inhibin directly acts on the anterior pituitary for negative feedback of FSH, while it also slightly influences the GnRH secretion from hypothalamus. In females, in addition to the negative feedback regulation by sex steroids, like in males, there also occurs positive feedback regulation at certain times. In females, the estradiol and progesterone act on the hypothalamus to inhibit the release of GnRH and on anterior pituitary to inhibit the release of gonadotropins, that is the negative feedback regulation. Thus, in major part of the reproductive cycle, the estradiol and progesterone act on the brain and anterior pituitary via negative feedback to inhibit the release of GnRH and gonadotropins. However, in primates during mid to late follicular stage and in rats on the day of proestrus with an estrous cycle of 4 to 5 days, the estrogen show positive feedback effect on GnRH release. This effect of estradiol causes a pre-ovulatory GnRH surge, which is followed by a pre-ovulatory LH surge, resulting in the release of ovum from the ovary and maturation of the corpus luteum. After the ovum has been released, the effect of ovarian sex steroids again becomes inhibitory on hypothalamus, that is, it again becomes the negative feedback mechanism until ovulation in the next cycle. In case of brain-pituitary-gonadal axis, the main site of feedback regulation by sex steroids is not GnRH neurons, as they express only estrogen receptor beta and not the other receptor like estrogen receptor alpha or androgen receptors or progesterone receptors. The presence of only ER beta does not satisfactorily explain the feedback regulation of GnRH secretion by estrogens, indicating that there are some other stimuli from central nervous system which also contribute in the regulation of GnRH cells. These stimuli may lie in hypothalamus or some other regions which contain sex steroid hormone receptors. Thus, the hypothalamic pituitary gonadal axis may more appropriately be called as the brain pituitary gonadal axis. The same logic may be applied to other axes, as the feedback regulation does not entirely depend on the hormones from the target cells only. Now, let us talk about the positive feedback regulation of oxytocin. Oxytocin is secreted by the dendrites and cell bodies of supraoptic and magnocellular neurons from paraventricular nucleus in hypothalamus into the posterior pituitary. Along with this, oxytocin may also be secreted by uterus, placenta and fetal membranes during parturition. During childbirth, when the uterine contractions start, more oxytocin is released, which stimulate more contractions, and as a result, still more oxytocin is released. Due to this, the strength of contractions increase, which help in propelling the baby out. Once the baby has been delivered, contractions stop and hence the release of oxytocin also stops. Thus, oxytocin secretion increases due to stimulation by neural signals from contracting uterus and cervix as the fetus passes through them. Certain drugs inhibit the secretion of oxytocin and thus slow down or stop the parturition. These drugs include microreceptor agonists like morphine and pethidine and kappa agonist. Another important role of oxytocin is in the release of milk in nursing mothers. Suckling of mother's breast by the baby results in the secretion of oxytocin in the mother's milk. This causes milk to flow in the mammary glands, and if the baby stops suckling, the milk is not released. The oxytocin receptors are expressed in lactotrophs in anterior pituitary. Together with prolactin, oxytocin helps in the ejection of milk from the mammary glands. The factors affecting the brain pituitary gonadal axis. The GnRH neurons are functionally regulated by several neurotransmitters and neurotrophic factors, reflecting that GnRH neurons are integrative. Neurotransmitters which provide input to GnRH neurons include GABA, glutamate, catecholamines and monoamines. Neuropeptides include CRH, beta endorphin, neurotensin, vasopressin, etc. Now, the next axis is the neuroendocrine axis for metabolism, which is also known as the brain pituitary thyroid axis. The brain pituitary thyroid axis is involved in the regulation of metabolism, which is needed for survival and development of the organisms. The rates of most of the intercellular reactions are regulated by thyroid hormones. The hormones which are a part of this axis are Hypothalamus secretes thyrotrophin releasing hormone, that is TRH, produced by the neurons in the paraventricular nucleus. Anterior pituitary secretes thyroid stimulating hormone TSH, and the thyroid gland secretes thyroxine and triiodothyronine. The large axons of the TRH neurons, like other hypothalamic neurons, terminate in the median eminence. The high capillary network of the median eminence carries TRH to the anterior pituitary. The receptors for TRH are located in the specialized cells in the anterior pituitary called as thyrotrophs, which when stimulated by TRH will secrete TSH. From the anterior pituitary, TSH is released in the general circulation and reaches its target organs, that is thyroid gland located in the neck, which has receptors for TSH. The thyroid gland secretes two potent hormones, that is thyroxine T4 and triiodothyronine, that is T3 in the general circulation. The target organs of T3 and T4 are located throughout the body. T3 and T4 are also involved in feedback regulation of TRH and TSH by acting on hypothalamus and anterior pituitary respectively. The brain pituitary thyroid axis is an example of the negative feedback regulation. The precise regulation of these hormones is important, as the lower or higher levels of these hormones may have drastic influence on the metabolism and other events of development. The thyroid hormones exist in two forms after being secreted from the thyroid gland: free form and the bound form, when they are bound to globulins. The free form of thyroid hormones, which constitutes less than 1% of the total thyroid hormones, participates in the negative feedback regulation by acting on TRH neurons, thyrotrophs, and also acting on the target tissues. The next axis is the neuroendocrine axis for lactation, also known as the lactotrophic axis. There is no releasing factor from hypothalamus for lactation, and only the anterior pituitary secretes the prolactin, which is a major regulator of lactation in mammals. Prolactin promotes the development of mammary glands and the production of milk. However, there is an inhibiting factor for lactation, that is dopamine, which is present in the hypothalamus. The prolactin is released by the lactotrophs in the anterior pituitary into the general circulation, from where prolactin goes to its target cells in the mammary gland and promotes the synthesis and release of milk into the alveoli of mammary glands when stimulated by suckling. Prolactin and oxytocin together are needed during the period of lactation. There are several factors which contribute to the secretion or inhibition of prolactin from anterior pituitary, like drugs which increase the dopamine levels in the pituitary portal capillaries influence the prolactin secretion. External application of dopamine influences the release of prolactin from anterior pituitary. The lactotrophs in the anterior pituitary contain receptors for dopamine, such that the increase or decrease in the portal dopamine is directly correlated with the changes in the secretion of prolactin from anterior pituitary. Though there is no true prolactin releasing hormone in the hypothalamus, but there are several factors like TRH, vasoactive intestinal peptide, and oxytocin which influence the stimulation of prolactin release. The next axis is the neuroendocrine axis for stress, the brain pituitary adrenal axis. Stress is an important physiological and psychological phenomenon to which the organisms must adapt or react properly in order to survive. There could be several reasons of stress, for example, escaping the predation, escaping the chronic stress of a disease, or escaping from the social influence. The response to stress is mediated by the brain pituitary adrenal axis, which works by secretion of following hormones. Like hypothalamus secretes corticotropin releasing hormone CRH, mainly from the medial parvocellular part of paraventricular nucleus and arginine vasopressin. Anterior pituitary secretes adrenocorticotropic hormone ACTH, beta lipotropin, 3-endorphin, catecholamines. Adrenal cortex secretes glucocorticoids, that is cortisol and corticosteroids, etc. The cell bodies of CRH neurons are located in the paraventricular nucleus of hypothalamus. CRH neurons receive input from various other neurons in the limbic system and the lower brain stem. The axons from CRH neurons in hypothalamus reach the external layer of median eminence, and from here the CRH neurons, they go to the anterior pituitary where they target the corticotrophs which secrete ACTH. Another hypothalamic hormone produced by magnocellular neurons in paraventricular nucleus is called as arginine vasopressin AVP or antidiuretic hormone or ADH, which also acts on the corticotrophs of anterior pituitary and shows a synergistic effect. The corticotrophs have receptors for both the hypothalamic hormones, that is CRH and AVP, and they respond by promoting the synthesis and release of ACTH from anterior pituitary. ACTH is released from the pituitary gland in the general circulation, from where it reaches its target organ, the adrenal gland, especially the adrenal cortex. The ACTH activates the adenylyl cyclase in cell membrane of adrenocortical cells which form cyclic AMP. Cyclic AMP induces the synthesis of adrenocortical hormones. Thus, adrenal cortex responds by secreting glucocorticoids. The glucocorticoids show negative feedback regulation of brain and pituitary and thus inhibit the synthesis of CRH and ACTH respectively. Glucocorticoids also inhibit the transcription of the precursor protein pro-opiomelanocortin POMC in the corticotrophs in the anterior pituitary. Thus, the brain pituitary adrenal axis is an example of a negative feedback regulation. The paraventricular nucleus is an important center as it gets input from various regions of the brain like the brain stem, midbrain, limbic system and other hypothalamic nuclei. Now, let us just talk about some factors which affect the CRH secretion from hypothalamus. There are several factors which may affect the secretion of CRH from hypothalamus like physiologic stress. Any type of mental or physical stress can increase the secretion of ACTH and cortisol. If there is any physical stress, the signal is transmitted via brain stem to hypothalamus and median eminence. Then CRH is secreted into the portal system in median eminence, which ultimately results in the secretion of cortisol in the blood. Similarly, if there is any mental stress, the signal is transmitted through limbic system, especially amygdala and hippocampus, which then transmit the signal to hypothalamus. Another factor is cortisol. Cortisol shows the direct negative feedback on hypothalamus and pituitary gland. Thus, cortisol decreases the secretion of CRH and ACTH respectively. Adrenal steroids use feedback loop to inhibit secretion of CRH and ACTH. In this type of feedback loop, there is a specific point which is called as steady state, where the secretion of releasing factors to stimulate the secretion of hormone is balanced by the end products to inhibit the secretion of the hormones. The steady state, however, changes over time in sync with the circadian rhythm. At some other times, more ACTH may be required to inhibit the release of more ACTH. Thus, let us summarize the chapter. Hypothalamus secretes several hormones which act on the respective target organs via several pathways which are called as brain pituitary target organ axis. Hypothalamus secretes various signals in the form of either hormones or neurotransmitters. The feedback regulation of neurohormones can occur at various levels like at the gene transcription level, translation level, the post-translation levels or even at the release of the hormones. The feedback regulation of the neurohormones is of two types: the positive feedback regulation, when the presence of high levels of hormones stimulate the secretion of more hormone, and the negative feedback regulation, when the presence of high levels of hormones inhibits the secretion of hormone. The hormonal signals from the hypothalamus are released into the hypothalamic hypophyseal portal system, a rich network of minute blood vessels in the median eminence just below the hypothalamus. From the median eminence, the hormonal signals go to the sinuses in the anterior pituitary. The somatotropic axis includes the secretion of hormones from hypothalamus which secrete growth hormone releasing hormone and growth hormone inhibiting hormone or somatostatin. Anterior pituitary secretes growth hormone which is also called as somatotrophin. The primary target organ, that is liver, secretes insulin like growth factor 1. Somatotropic axis is an example of negative feedback regulation, as excess of growth hormone and IGF-1 inhibit the release of hormones from hypothalamus and anterior pituitary. The brain-pituitary-gonadal axis secretes the hormones as follows. The hypothalamus and the preoptic area secretes gonadotropin releasing hormone, also called as luteinizing hormone releasing hormone or LHRH. The anterior pituitary secretes gonadotropins, that is luteinizing hormone and follicular stimulating hormone. The target organ, that is gonads, testes in males and ovary in females, they secrete sex steroid hormones like estrogens, progestins and androgens. During most of the time, this axis shows negative feedback regulation, as sex steroids inhibit the secretion of anterior pituitary and hypothalamus. But in primates, during pre-ovulatory stages, the sex steroids adopt positive feedback mechanism to increase the level of LH to cause LH surge, which is required for ovulation. After ovulation, the axis again adopts the negative feedback regulation. The brain pituitary thyroid axis secretes the following hormones. Hypothalamus secretes thyrotropin releasing hormone, that is TRH, which is produced by the neurons in the paraventricular nucleus. Anterior pituitary secretes thyroid stimulating hormone TSH, and the thyroid gland secretes thyroxine and triiodothyronine. This axis follows the typical negative feedback mechanism for regulating the secretion of TRH and TSH. The thyroid hormones, when present in higher levels in the tissues, exert an inhibitory effect on anterior pituitary and hypothalamus. The lactotrophic axis does not have any releasing hormone from the hypothalamus. However, hypothalamus has dopamine which inhibits the secretion of prolactin from anterior pituitary. Suckling by the baby stimulates the lactotrophs in the anterior pituitary to release prolactin, which acts on the mammary gland and stimulates the production and secretion of milk. Prolactin and oxytocin together stimulate the release of milk formation in the mammary gland. In the absence of suckling stimulus, dopamine inhibits the secretion of prolactin from anterior pituitary. The brain pituitary adrenal axis secretes the following hormone. Hypothalamus secretes corticotropin releasing hormone CRH, mainly from medial parvocellular part of paraventricular nucleus, and arginine vasopressin or AVP. The anterior pituitary secretes adrenocorticotropic hormone, that is ACTH, 8 lipotropin, 3 endorphin catecholamines, and adrenal cortex secretes glucocorticoids, especially cortisol and corticosterone. This axis is also an example of a typical negative feedback regulation. The glucocorticoids show inhibitory effects on the anterior pituitary and hypothalamus when the glucocorticoid levels become higher. Thank you.